Ma'am,
Yes, Aditya, tell me.
ma'am, just wanted to confirm that you have uploaded this PPT on the sheet, right?
On Flipper? No.
No, the sheet, Excel sheet, spreadsheet of Academy.
Okay, let me check. No,
Grade
I
9 and 10
haven't.
content.
Acha, that one. No, it's not there. So once I
Okay,
finish the class, which is once we are done on Thursday, right? I'll share it on our WhatsApp group only.
okay, ma'am.
Okay, okay, okay. So let's start. Yeah, I think Divya's also joined, right? Let's start. Right. So basic introduction, right? Okay. What is reproduction? It is basically the production of new organisms from existing organism of a species that ensures continuity of population of that species, right? Okay, so basically reproduction is nothing but a way to continue the species on, right? Oh, I was supposed to ask you this as a question. I pressed the slide and all the points came. Okay, not to worry. Okay, so in term one, you all did this chapter called life processes, right? In that you studied about various life processes like what all did you study?
Nutrition,
Nutrition,
respiration,
respiration,
transportation, excretion.
excretion.
Correct, yeah, nutrition. So basically you've done everything from digestion, right? Assimilation, absorption, transportation. Okay, I'm just writing in short. And excretion. Right? So these are the life processes which are absolutely essential for growth and development. Right? Okay. Now reproduction is also a life process, but this is not necessary for growth and development in general. Right? But its main purpose is so that the species continue. Okay. Yeah. So this is not for growth and development, but it is a life process which comes under uh, extending the species. Okay, so why is it necessary or why is it essential is continuation of life, addition of new species, replacement of dead organisms, transfer, adaptations and variations from one generation to another. So now this point and this point, which is basically the second point and the fourth point, we'll be looking a bit in detail when we do heredity. Okay, that time, since there is some basics of evolution also which is included, I will tell you how reproduction uh, leads to addition of new species and what does this actually mean, right? This we might even cover variation today also a bit, but yeah, more in detail when we do heredity, right? So continuation of life, basically, yeah, you would like your species to continue to exist, right? You would not like to be wiped out completely. Addition of new species generally occurs when there is a variation or when there is a mixture of genes, right? That's how evolution actually happened, right? If you look at it, earlier there were cavemen, right? Even if you even consider just humans, we have been through a whole cycle of evolution, right? Or if you look at life, the beginning of life to now, it is said that we've all evolved from one organism, right? Does anybody know which organism did we all come from?
Apes or monkeys?
Okay, that would be humans. Okay. What about if you go really, really back?
Dinosaurs.
No. Even before that.
Plants.
Guess. Any guesses you can guess.
Will it be unicellular animals?
No. Okay. So let me tell you, it is believed that we've all come from fishes. So fishes were the initial life forms on the planet. One of the very first life forms on Earth were the fishes or aquatic animals, if you could just categorize them, generalize them. Okay. So what happened was the fishes or the aquatic animals then started moving on to the land, and that's how all the land animals came into existence. So it's actually come out from a fish. Okay, yeah. If you're interested in evolution, if you're interested in those kind of studies, there are a lot of resources, right? There is this person called Charles Darwin. He's said to be the father of evolution, right? So he has a lot of theories and a lot of studies which he's done to show, how species came into being and etc. etc. In that, you will get all these interesting, uh, yeah, information. Okay. Huh. And one more thing I wanted to tell you before we started the chapter was these terms. So these three terms become very, very important when you do this chapter and when you do heredity, right? Because if you are confused between what is DNA, what is genes, what is chromosomes, right? So understanding terms or understanding them when they are used in sentences, explanations becomes difficult, right? So I've just made given a simple diagram here so that you can understand the difference between the three. Once you're clear with the difference between the three, I don't think you'll go wrong anytime. Like in heredity, if you're doing heredity now, you might be coming across words like traits, right? What is the meaning of trait versus what is genes, right? All those words are new, so we'll just get this concept clear. Okay. So DNA, I think all of you know the full form. What is the full form of DNA?
Deoxyribonucleic acid.
Deoxyribonucleic acid.
Yes, awesome. So DNA stands for deoxyribonucleic acid, right? So DNA is a type of nucleic acid. So basically, DNA is present in the nucleus of a cell. Okay. And how is it present? It is coiled. It is a highly coiled structure. So if you look from here, right, there is this ladder-like structure of the DNA which gets coiled multiple number of times to form this structure, which is known as a chromosome, and ribbon-shaped X-like structure. Okay, and this is so chromosomes are then found in the nucleus floating like this. Okay, like here. Okay, this is the nucleus of the cell, right? So DNA is in the form of chromosomes inside the cell. Okay. So now what is the difference between genes and DNA? As you can see in the diagram here, DNA would be the entire stretch, or DNA is basically what makes up the chromosome. Or you could say that the chromosome is comprised of DNA. Okay. Whereas what is a gene is a part of a DNA. Okay. It could be any part or any segment, right? The correct word would be segment here, right? Any length of the DNA which performs a particular function, or if we, I mean, when we go to heredity, we'll learn it better that genes are basically units of heredity. They carry important pieces of information for some particular task, right? So this could be a gene for, let's say, eye color. Okay. So eye color is coded by this much segment of the DNA. So that much segment is then called a gene. Okay, so this is the difference between the three terms. DNA is what makes up the chromosome, right? And gene is nothing but a segment of the DNA which performs a particular function. Okay, okay, let's go ahead. Okay, now there is this huge paragraph which was there in your textbook itself. Though it is not a really important one, but yeah, if you read it, you might get confused as to what are they saying. So I thought I'll just include it here. What are they saying? Right? So they've asked you a question saying that do organisms create exact copies of themselves? right? What do you think? Do organisms create exact copies of
So in
themselves?
asexual reproduction, yeah, they may create exact copies, but in sexual reproduction, there's always a variation.
Correct. Yeah. Right. Okay. So basically, always they will not be creating exact copies of themselves. If that were
Because
true,
faults in DNA copy.
faults in DNA. Yes, correct. Okay. If that were true, we'll all be looking the same, right? There would be just same copies of the, there would be same copies of one person going around, right? If you take plants also, there would be just same copies of the same plant everywhere, right? There's a little bit variation involved. Okay. So organisms look similar because body designs are similar. And if body designs are to be similar, the blueprints for these designs should be similar. Right? Basically, what they're trying to say is body designs are similar, as in, uh, if you take humans for example, right? We are supposed to have two eyes, one nose, uh, one mouth with maybe 30 to 32 teeth, right? Ranging whatever, 2 years, right? Hair, etc. Okay. Yeah, two arms. Forgive my drawing. Two legs. Okay. Yeah. Something like this. So this is the basic blueprint. Okay. So for example, if human basic blueprint is, let's say this. Okay. With two arms and two legs. So whenever you make a copy, or basically whenever reproduction occurs, it has to make sure that this basic blueprint is followed, which means you must have 2 ears, two eyes, one nose, one mouth, two arms, and two legs, right? So this is the basic blueprint which they are talking about, right? However, what happens is when this copies is being made, so basically how does this information, or how does a copy of a blueprint is made, is when the DNA replicates, right? DNA replicates to create copies. Okay. So that is not a foolproof method, like Puneet mentioned, right? There are errors during DNA. So DNA copy. So you might get somebody with the same framework, but maybe you might have, let's say, blue color eyes. Okay. You might have a slightly crooked nose. Okay. You might have less number of teeth. Okay. Yeah. Something like that. Okay. Shorter arms, longer legs, all those things become the changes in the blueprints. This is just a simple way of putting it across. Okay. So DNA in the cell nucleus is the information source for making proteins, right? So what happens basically is this DNA in each and every cell which is there, right? It makes copies of itself, then it turns into RNA, and the RNA finally turns into proteins. Okay, this is a complete cycle which keeps happening in the cell. And this protein is what gets, it's a term called expressed. Okay, expressed as in this protein would show up, so that then you would know that you have blue eyes. Okay, so the DNA coding for eye color, let's say. Okay, it first will make a copy of itself, then it would turn into RNA, then it would turn into proteins. I'm putting it in a simple format. It's not a single stepwise like this. There are lot number of steps involved in DNA making a copy of itself, DNA translating into RNA. The term is called translating. Okay, and RNA translating to proteins, right? There are a lot of steps involved in this. But in short, if I were to explain, this is how it happens. So when the proteins get expressed, the person will have blue eyes. Okay, or you will see that, yeah, there is a person with blue eyes among you, or something like that, right? Same for any other eye color, same for anything you take in your body, right? Every small thing. There are proteins coding for specific enzymes which you have, right? How your skin texture will be, how your nose will be, how your teeth will be, etc. For every small thing, this is how the entire procedure looks like. So what happens is when this process happens, because there are chemical reactions basically, There is no foolproof method. There are faults which happen. Sometimes while making a copy, some part of the DNA gets deleted, right? Some part gets duplicated, or some part goes uncoded. There can be a lot of things which happen which lead to mutations, what are called mutations, the technical term. Or in simple terms, you could call them errors, right? Which could lead to a lot of different things. One example I could give you would be having, let's say, 6 fingers instead of 5, right? 2 thumbs. It's just an example. Okay. This is a case where some reaction or this entire process has gone faulty, right? There's some fault which has occurred in this, which is why you end up with 6 fingers instead of 5, right? Okay. So this is what the entire paragraph in the textbook is trying to convey, that basically you will not have the same blueprint. You will not have the exact same body design, right? You'll have a similar one. Okay. Yeah. No biochemical reaction is absolutely reliable, right? Which means it's prone to errors, basically. Okay. Which means copying DNA will have some variations, right? So these changes, basically the changes which happen, are called as variations. Okay. They don't have to be that drastic, like a 6th finger or an additional thumb, something like that. Okay. There could be very small, minute ones also, which go unnoticed. Okay. That is just an example I gave so that you can relate to, okay, yeah, there is something like this or things like that, right? There could be very, very small changes also which happen, right? Which go unnoticed because they do not really impact the individual. But in short, there are changes which have happened. Okay, so those changes are what are called variations in technical terms, or in when you talk about heredity and evolution, that's where the word variation would be a technical term. Okay. As a result, DNA copies generated will be similar but may not be identical to the original, right? You can take a good example of twins also. Okay. Right? Twins, we have two different type of twins. Do you know what are they called?
Identical and non-identical.
Correct. So you have something called identical twins and you have something called non-identical or fraternal twins. Okay. Right? So if you have seen, even if you see identical twins, right, there are a lot of features which are different in them. Have you noticed? Do you have any friends who are
Yeah,
identical
like
twins?
if you look overall, they're same, but then if you know them from a long time, then you can check the difference between who's who.
It's
Exactly.
mostly behavioral.
Behavioral, definitely. Behavioral would be definite this, right? But I was talking of more of, you could say, physical appearance or something which you could,
Eyesight.
Yes,
yeah,
it's just different.
notice. Correct. Eyesight, yeah, Punitya, you could use that as an example. Right? So if you look at twins, okay, identical twins, right? In them also there are observable differences which you can see, right? Although overall they might look the same, they might have maybe the same kind of facial features, or maybe they must be of the same height, okay, but there will be some genes which are different, right? So this is why it happens, even though they are uh, from the same cell, right? They've developed from the same initial cell. There will be a lot of differences because of this, right? Okay. Okay, yeah. So here only they are speaking of why variation is necessary, why this change is necessary, and why we don't really go for a copy paste method, right? Like in terms of asexual reproduction, where the exact same copy happens, there is no chance of having variation, right? So here are the points why you need variation. Again, this is given in the form of a paragraph in the textbook. I just took it right from there so that I could explain what it means, right? Again, this part is also a little bit connected with evolution, because you need to understand that to understand why variation is important. Okay, so populations of organisms fit or fill well-defined places, or niches they are called, in the ecosystem, using their ability to reproduce. So in the ecosystem, every organism has a fixed place, or you know, you could talk about, let's say, uh, something like the food web. You've come across the food web, right? So every organism occupies a different level in the food web or the food chain, right? Suppose, let's say, insect to frog to, let's say, uh, I don't know who eats a frog. I don't know. Wait, wrong example.
Snake.
Snake. Okay, let's take snake. To, let's say, take eagle or hawk or something like that. Okay, or let me take a better example. Hmm, okay. Fish to, let's say, large fish, to, let's say, some animal, to human finally. Okay, so this is what is our food chain, right? So here they occupy a specific level in the food chain, right? All these organisms. Similarly, when you talk about ecosystem in general, there are these places, or they are called niches, okay, where each organism has a specific niche, or it has a specific location of their own. Okay, that's what a niche is. If a population of reproducing organisms were suited to a particular niche, and if the niche was drastically altered, the population could be wiped out. Okay, for example, they've given you a good example here down of this bacteria, right? Suppose there is a bacteria surviving in temperate waters. Temperate would be referring to the climate. Okay, the climate is moderate climate, that's called a temperate climate. You have a temperate zone also, I think, in you might have studied in geography. Okay, so basically moderate climate basically is what it means. Okay, and if the water temperature was increased due to global warming, most of the bacteria would die, right? But the few variants resistant to heat would survive and grow further. So what happens is, because of variation, there would be chances that some of the bacteria would get heat resistant, right? And those would be the one who would be able to survive this change. This is just an example they have taken. We can take another example of what is happening right now. You can take the example of COVID.
Omicron.
Very beautiful example it is for the same thing, right? So what happened is, it started as something called SARS-CoV, right? COVID-19. It started off as one variant, and that variant, it divided, and it gave rise to a lot of other variants which were called mutations. If you see in the news, you might have seen this word a lot, mutations. It has mutated and given rise to alpha, then there was beta, gamma, delta, and now finally Omicron, right? So this is what has happened. So COVID-19 gave rise to a lot of uh, it has a rapid multiplying rate. So it gave rise to a lot of new, what should I say, new individuals. I'm trying to say that basically it reproduced, right? And because of variations, it realized, it came up with some of them which had better chances of survival. Okay, and these variants became deadly to us, right? That's how it works. It's not always for the good. Sometimes these variations can cause uh, detrimental effects as well, like in this case. Okay, so when they talk about COVID, they will be always talking about how deadly it is or how harmful the mutation is. right? So out of which Omicron and Delta were the two successful mutations or the two successful variations which seem to be causing a lot of infection in humans because they were able to stay in the lungs better, or they were able to how should I put it? They were able to stay inside the human and were able to multiply better, right? So you can take this also as an example of variation, right? It's not an exact clear variation example, it's a related one, right? So it gave rise to a lot of individuals. Let's call them 1, 2, 3, alpha, beta, gamma, 4. Okay. So out of that, maybe 1, 2, 3, 4 were quite similar. But there were variations in alpha, there were variations in beta, gamma, which we finally identified, right? Something like that. There are a lot of these examples. When you study evolution, you might actually come across a lot of examples where variations has led to some species continuing further. Okay. So this is that entire paragraph in the textbook which talks about importance of variation, right? So basically you just need to know this point. This would be sufficient, right? So if everybody had the uh, same body design or the exact same replica, then there could be chances that the population could be wiped out, right? If there was any alteration. But due to variation, some of them would survive and hence the species would continue. Okay, that's the gist of it. Okay. Okay, now we come to the main part of the chapter, which is the types of reproduction. Right, first one is asexual reproduction. These are some of the features. Again, sometimes it's asked as a direct question, so I just included it as a separate point, right? The main features are only one organism is involved, right? New individuals are genetically identical to the parents, which is what we were talking about till now. It is a rapid mode of multiplication, important point. Yeah, this is linked to the first two, right? No gametes, therefore no fertilization. Therefore, it is a rapid. Therefore, the individuals will be genetically identical to the parents, right? Okay. This is just you can take this down if you want, or you can take it down when I share the PPT. Anything is fine. Just make sure you have this somewhere as features, because sometimes they ask these questions also. What are the features of asexual reproduction? Oh yeah, genetically identical. Oh, it's come twice. It's the same point, just repeated twice. Okay. Yeah, so now we'll talk about the different ways of asexual reproduction. There are some of them. Okay, one is fission. Do you know the others? Which are they?
Fragmentation, budding,
Vegetative propagation.
Right. And
Spore formation.
spore formation,
and one more missing.
Regeneration.
Yes, correct. Good, good. So you all are up to date with the chapter. That's good. Yeah, so these are the 6 types which are there for you to study right now, right? Out of which this becomes for plants, right? One exception, which is specifically for plants. Spores is for fungi, right? Oh, what happened to my handwriting? Yeah, fungi, right? Fission is for unicellular.
Amoeba.
Amoeba, right? Fragmentation.
Miss, but there is multi.
Sorry, there is.
No, no, not.
Okay, right. So out of which these can be either by unicellular or by small animals, right? There are a lot of examples, etc. All regeneration and these three. Vegetative is specifically for plants. This becomes a whole huge area by itself because there are a lot of ways of doing vegetative propagation.
Root,
Spore
stem, and leaf.
formation, root, stem, correct. Yes, yes. Spore formation is specifically only for fungi, fungus, right? All the different types of fungi, they form spores, and that's how they propagate. Regeneration is ideally a type of what do you say, body repair mechanism. It's not generally a way of reproducing. It's just like a last resort kind of thing.
Growth.
Growth, yes. It's more for growth. Okay. Yeah, so these are our 6 types which you have, out of which vegetative propagation is a field in itself. Why? Because people, there is this, there is a very small paragraph given in the textbook, but it's a huge field, right? So people do their what do you say? They do their graduation in tissue culture, things like that. It's a very, very huge field.
Microbiology.
Microbiology is different. Microbiology would be study of all the microorganisms. So that
All
is a
vaccines and
that is a different field in itself. In that you will be studying all about their cell walls, how do they grow, how do you cultivate them, how do you use them to get useful compounds. So now I think you might have seen there are a few people who have come up with microbes which can clean the oil spills in the ocean, some things like that. So that and all you will be studying in microbiology. Tissue culture, what you do is you take up different plants, right? You can take, let's say, the root of maybe potato and the shoot of
tomato.
tomato. Yes.
Tomato.
Yeah, and make something called a pomato. Yeah, they discontinued it because it was not of any use, right? This is just an example I'm giving you. This is how they are looking to enhance or enrich or take the good from the potato and take the good from the tomato and create hybrids, things like that. Or if you would have heard, there is something called uh, golden rice. I think you will come across it later, right? So golden rice is basically they fortified the rice with vitamin A.
For that it was they were having some problem. Eyes was right.
I think this was during the agricultural revolution.
There
Yeah,
was a
for
deficiency of of vitamin A in a
yes.
region, and the region's main diet was rice. So they modified rice so that they could all survive.
Yes, correct. They modified rice so that there would be, and more of malnourishment also was going on. So they fortified rice with vitamin A and they called it golden rice. So all this comes under tissue culture, right? This would be a part of it, a sub part. More would be how would you propagate different plants using their parts of them, right? Using just a stem or just a leaf, and how do you mix and match? Right? Those kind of things come in tissue culture.
Tissue culture is a part of vegetative propagation.
Yes, it is a part of very important part of vegetative propagation. Yes. Okay. So fission is, just points here. Process by which unicellular organism splits into two or more than two, right? When it is two, it is called binary. Am I audible? Some issue? Or is there some
Yes.
issue? Audible now? Hmm. And more than two would be multiple fission, right? More than two. And this is two. Bi refers to two. You can always remember like that. Bi is always two. Binary. What other word is there? Suddenly I can't think of it. Okay. Let it be.
Die.
Di is also two.
Binomial.
Correct. Binomial. binomial. You have something called binary, which you might come across when you do computers or coding, decoding kind
Yes,
of.
0 and 1.
Zeros and
Zero.
ones.
Correct. Yeah. So those are. So bi basically means two. So wherever you see a word like binary would be two. Okay. Multiple three, yeah, you know. Most common and simplest method, and it is seen in which organisms is like yeast, bacteria, and protozoa. Okay, these are the
Ma'am,
important.
yeast is budding also, right? So if they ask what is the product more for reproduction
Yeast
in
is
yeast,
mainly budding, right?
Yeah, both actually, but they won't ask you this question. They won't ask which both. What they might ask maybe is one organism which has both. They could frame a question like that. Like which organism can reproduce via fission and budding. Then you can say yeast. Okay, they won't ask the other way around. So yeast also, it's not actually just one organism. There are a lot of types of yeast. There are subspecies, right? So there's something called baker's yeast, that is Saccharomyces cerevisiae. Those are all Latin names, right? There's something called brewer's yeast. Okay, so baker's yeast is the one you might have you might have at your house, or ones you use to make bread,
Put it
etc.
in bread items
Correct.
or
Yes.
pizza base.
Yes, all of them. So that one is called baker's yeast. Okay, it's called as, if you want the name, it is, this is the scientific name, Saccharomyces cerevisiae, right? They won't ask you this, I'm just giving you information, right? Then there is another type of yeast called brewer's yeast. This is used to make alcohol. All the alcohol drinks, beverages use this yeast. Then there are a lot other ones also, like some of them are used specifically only for ethanol production. And for other byproducts, they are engineered, etc. So there are a lot of subcategories of yeast. They did not want to tell you which one at this level. They did not want to give you that much information, so they just put yeast both places. Okay, yeah. So bacteria, yeast, and protozoans, remember that is fission. So there are two types, binary and multiple. We have both here. Binary fission, main example is amoeba, right? The parent organism divides into two equal and identical daughter organisms. Okay, yeah. So here you need to remember the steps. The steps would be, first nucleus divides, second cytoplasm divides, right? And third you have daughter cells formed. Okay, so it will go in this order. First nucleus, then cytoplasm, then daughter amoeba. Okay. Okay. Yeah. I don't think there's much to explain in this. You remember pseudopodia, right? They are called false feet. Pseudo is false. These are basically false feet. Or these extensions which you see here, all these are pseudopodia. Okay. So yeah, pseudopodia comes in, then the nucleus divides, and then there will be, where the nucleus divides itself, there will be like a groove kind of thing forming, so that the cytoplasm gets pinched off, and you will end up with two daughter amoeba. So when you make the diagram, just make sure you're showing nucleus division and you're showing cytoplasm getting pinched off and two daughter amoeba form. This is important. It's not much to label in it, right? Okay. And you have another example. So this main reason why they gave you one more example was amoeba is an organism which doesn't have a particular shape, right? It's shapeless basically. It has no shape, and if you look at it, it can divide from anywhere, right? So there is no start point, end point, or there's no center point ki acha yahan se hi hoga, or the cytoplasm will get pinched from here itself. There's nothing like that, right? So they wanted to, I guess they wanted to make it clear that That is not what happens in all the organisms. That's what happens mainly in amoeba and paramecium, right? Amoeba more shapeless, paramecium has a little bit more shape at least, right? And when you look at other organisms like leishmania, okay, leishmania is an organism which causes the disease Kala Azar, right? It's a type of fever, deadly fever, right? They have this kind of structure, so it looks something like a tag, you could say, very weird looking tag. Okay, they have a whip-like structure at the end of the cell. So what happens when organisms have a definite structure is that time there is a fixed place that yes, there will be division happening from here itself, right? Now the place is fixed. So the groove will be formed from here and then the cytoplasm will be pinched from here and you will end up with two daughter organisms. This is not frequently asked. Amoeba is frequently asked. Leishmania, they usually ask like they'll tell you which organism causes Kala Azar and they, uh, you are supposed to write the spelling of Leishmania. So it has a tricky spelling, right? Okay. And there's not much information about it. So that's the only question which usually comes. But it's good to have knowledge of this diagram as well, right? Yeah, because they can just tell you which organism is this, identify. So you should know, yeah, leishmania. Okay, and what is multiple fission? Multiple fission, you have the example of Plasmodium, which is the malarial parasite, right? It causes malaria. Okay. Multiple fission is parent organism divides into many identical daughter cells at the same time. Yeah, I have a video of all this somewhere once we complete up to vegetative propagation. I'll show you the video. So all of this is there in that, there in short and nicely it's put up, right? Okay. Can be seen in Plasmodium and I've just added one more example here, just additional resource. Monocystis. So parent cell will divide into many daughter cells and then that cyst breaks and the daughter cells are released, right? Again, not much to explain. Next, we have fragmentation.
Just can you go 1 second? Example.
Here.
Thank you.
Okay. Okay, next we have fragmentation, right? Common example, Spirogyra. I think earlier they used to have this Spirogyra image also included. Now I think they took it off. Oh, this is just a question. Okay. So organisms on maturation break into smaller pieces. Maturation basically when they become adult stage or when they have achieved some amount of required growth, right? They break into smaller pieces and each piece or fragment can grow into a new organism. And this is seen in organisms like Spirogyra and sea anemones. So Spirogyra basically has something like this. It has this kind of structure. This diagram used to be there in the textbook. And then it will break into fragments and suppose something like this and one more. Right, and each of them then can form a new organism itself. Here one more thing I wanted to tell you was the difference between these two words. Can anybody tell me, do you know the difference between these two words? What is a segment and what is a fragment?
It can be fragments or two different pieces formed from a single piece or segment can be, they are not different, they are attached to the same thing, but then they are differentiations. Like
Fragment can
Okay.
be called a part of something.
Okay. Okay. Quite close. Quite close. Both of you quite close. Good. Yeah. So segment is basically, segment is you could say a lengthwise section, right? Suppose I make a line. Okay. Oh, this became maths now. Okay. If I say this is a line and I call this a segment, it's basically a lengthwise part of it, right? So this becomes a line segment, right? Whereas fragment is basically, like you all said correctly, broken piece. So segment is a lengthwise part of something. Okay, for example, if we go back, I was talking about DNA, right? In the first slide, we spoke about DNA. Let me draw it properly, right? This is something like DNA. So gene, if I say this is a gene, this is a segment of the DNA, right? It's called a gene segment actually. It's called a gene segment, right? So it is a lengthwise part of the DNA, which is a gene segment, right? But if I were to break this down and if I were to make a piece of it, if I broke the DNA down and I got out this much piece, then I would call this a fragment of the DNA. Right now, this would not be a gene. This would be just a fragment of DNA, right? Again, I thought these two words will keep coming up again here. So I thought you should know the difference, right? In case you don't know, you can write it down or you can write any example which you find it easy to remember. Okay. Yeah, so this is observed in multicellular organisms, right? Again, important. Fission is seen in unicellular organism. Unicellular is nothing but single-celled organism, right? We have done all this in 9th standard. Multicellular is many cell, right? So this is seen in multicellular organisms with a simple body structure. Examples are Spirogyra and sea anemone. Sea anemone is also a creature of the sea. Yeah, I can't explain it in a simpler way. Okay, yeah. So when does this happen is usually you need favorable conditions, right? Even if you take the example of seed, if you leave a seed outside like that, it will not start germinating, isn't it? Only when you give it proper sunlight, when you give it enough amount of moisture, or when you place it in the soil, right? Only then will the seed start to germinate. Similarly, it happens for all uh, reproduction you consider, right? You need favorable conditions. Those conditions might be different for different organisms. Like plants might need water, sunlight, soil. Uh, Spores might need drier conditions, or they might need more more sunlight, something like that, right? So it is different in different organisms, but the favorable condition idea will remain the same, right? Only then will reproduction occur, because if the conditions are not favorable, it definitely would mean the species would not be able to survive, right? Okay. So occurs under favorable conditions of moisture, temperature, and nutrient availability. Okay. Yeah, so this is a question for you. Can fragmentation work for all multicellular organisms? What do you think?
no.
No, in our case only it is not working
Correct. Good, good. Yeah.
because sometimes it's multicellular organisms. If the part is cut off, it becomes dead.
Not exactly. That is not the reason.
So
Uh,
all
We have
organisms are not small and symmetrical, right? For Spirogyra, you can just cut in the middle and then there are two pieces. For complex organisms like humans, you can't go with fragmentation.
okay, okay. But yeah, that would be basically what is already known, right? I was asking the reason why they would not prefer fragmentation. Why would it not work? One main important thing you are missing. Close.
Ma'am,
Yeah,
is it
Mansi is close.
ma'am, is it because like we have different organ systems and different places where certain functions happen?
Yes.
So if we like just split, then it won't work that way.
Again, okay. If you're saying split, okay, close. So more importantly, we have, I agree with your
Variation.
first point that we have different organ systems, right? And each organs has or each organ has a specific function, right? But more importantly, if you split that part and it will not grow, say zyada, we already have a definite organ for reproduction also as well, right? So we don't need to resort to this measure. Okay, so that would be the more apt reason. So like you all said, that is correct. First point would be multicellular organism. As you take the bigger animal, the complexity would increase, right? So if your complexity is increased, you have organ systems, you have complex organ system and functions, etc. Right? And the second point would be that since we have all that, we have dedicated organs for reproduction, right? This is the reason. Okay.
Also,
Whereas
can we
when
say
you
that in case of smaller organism like Spirogyra, even though it may be multicellular, but it was reproducing asexually, so fragmentation is possible.
uh, yeah, it is possible in multicellular organisms, but it's not possible for all of them. So this will work for ones which have simple body structure, which don't really have this organ system bifurcation, right? So they don't have any particular uh, system to go about it. So yeah, they resort to measures like fragmentation, regeneration, budding, etc. Okay. Okay, so this is the answer. In case anybody wants, you can take it down. Next one we have spore formation. Spore formation again will occur in multicellular organisms, right? More specifically fungi. Yeah, what do you need to know is this diagram and some terms, right? New terms, which is sporangia, spores, etc. So what are sporangia? Basically blob on a stick. They've just described this, right? So this structure looking like this is called a sporangia. Blob on a stick like structures. The sporangia are involved in reproduction. They contain the spores which develop into new individuals, right? And these spores are covered by thick walls that protect them until they come into contact with another moist surface and can begin to grow, right? Same thing, you can always take an example of seed, right? And you can you can take and make an analogy and look at it like that. So when you keep a seed outside, it doesn't germinate. And the same thing, the spores also, when you keep them like that, they won't germinate. Only when they have uh, favorable conditions, in this case would be moist surface, right? Only then the spores will be released like this. Here they have shown you how they get released, and then they will begin to grow. Okay. So this is all you need to know about spore formation. These stalks are called hyphae. Sometimes earlier they used to be a part of the diagram, so I've just included those also. Not asked, but still good to know. The stalks are called hyphae. The entire structure is called a sporangia, right? They have a hard covering, thick wall, and inside that the spores are. Okay. Example would be rhizopus. Common example if you were to take would be rhizopus, or you could also say bread mold. Okay. Okay, now we come to regeneration. Regeneration is basically, yeah, development of a new organism from a broken or cut part of the parent organism, right? For example, simple animals like Hydra and Planaria. Here you have two examples. One is Hydra.
In Hydra also we have budding, right?
Hydra also you have two, budding and regeneration, both. Correct. Hydra and Planaria, they can be cut into any number of pieces, right? As they have cut here. And each piece can grow into a complete organism. That is important, right? Usually what happens when you read this, you register that one piece grows into the complete organism, but it's actually all the pieces can grow into a complete organism, right? Okay. And how does this happen? They've
So
given
for
you
regeneration, only in case of few animal like planaria, it is like a mode of complete reproduction, right? In
yes.
some cases like starfish, it's not a mode of reproduction, but if one hand or one part of the starfish goes, it can regenerate.
Yes, correct. So regeneration is like a last resort for reproduction. Okay, it is not the first choice. It is a kind of a last resort kind of thing. Whereas the first choice for them would be more of fragmentation, budding, etc. Right. So if you notice, even planaria will have its own uh, system, either budding or something else. Right. But regeneration is like a last resort where when there is you could say when there is favorable conditions, or when the species need to propagate, yeah, they can use regeneration to continue this species. What Neki was saying, we have a lot of examples for that. One is starfish, where it is used as repair mode mostly, right? Regeneration as a repair mode would be starfish, lizard, right? Lizard's tail. I don't know if you've seen, if you cut the, yeah, lizard's tail.
Heard about it, don't want to see.
Yeah, okay. Yeah, so basically, if you accidentally pinch off a lizard's tail, it grows a new tail. Okay. Yeah,
Just what
Mansi.
is the difference between regeneration and fragmentation?
Okay. Difference between regeneration and fragmentation. Good question. So they are quite similar, but in regeneration, what happens is you can have a lot of parts, or uh, each part can grow. Whereas in fragmentation, there would be fixed places where the fragments can occur. Right? Regeneration also, there would be fixed places, but like I said, regeneration actually occurs through this route. It would start off as a repair, but then they would resort to developing the complete individual itself. Whereas fragmentation is, it doesn't start off as repair, it starts off as a mode of reproduction itself. That would be one of the major differences.
Ma'am,
Yes.
ma'am, do earthworms regenerate or they use fragmentation?
Uh, Neither actually. They have a complete their own huge reproductive system in their own this. So earthworms are what are called as hermaphrodites. I don't know if you've heard the term. Okay. I think you are talking of is when some part of the earthworm is cut off, it can regenerate. Is that what you're asking?
Yeah, ma'am.
So that again would come under repair then. So earthworms, they have their own complete, very complex reproductive system, and what do you say? One key factor of earthworms is that both the male parts and the female parts are present on the same earthworm itself, right? That's why they are called hermaphrodites, which means they have both of them in the same organism itself, and yeah, so that's how they would reproduce. They have organs for reproduction. Okay, they use regeneration for repair. So yeah, regeneration, basically it would start off as repair only in rare cases, or when the conditions are very favorable, or it feels like, yeah, this would help the species propagate, it would go ahead as a mode of reproduction, right? Okay. Yeah, I think Mansi gave a good example of regeneration in humans also, right? I don't know if you heard about it.
Ma'am, there can be fission also in humans. Like during reproduction, once a zygote is formed, after that, it
Uh,
creates many cells, right?
yeah, but that won't be called fission. That is called mitosis. That is a different thing. This is a different concept. What you are saying is called mitosis, where the cell divides, right, into two
Yeah.
daughter cells. Yeah, that is a complete different process, and it is known as mitosis. Fission is different because fission is occurring only in unicellular organisms. They have termed it fission because it occurs only in unicellular organisms. When it occurs in complex organisms or in humans, it is called mitosis, right?
But
Because
the
it's
process is same.
similar, not same. I would say
Okay.
similar because in mitosis, yes, the nucleus first divides, then the cytoplasm divides, but there are a lot of steps involved in nuclear division, and you need something called centromere, right? I don't know if you remember from grade 9 cell chapter. In the cell, there are these small structures called centromeres floating around, right? You need those for doing mitosis. So all those things are not required in fission, right? If you notice fission, it just happens by default. You don't require complex machinery. Okay, that's how they would be different. Okay. Coming back, I was saying there is a regeneration ability which humans also have, and the only organ which is capable of doing that is a liver, right? So if you, that's how liver transplants actually are done, right? So you can give out some part of your liver, you can donate some part of your liver, and when you give it, like, so the leftover liver in your body will regenerate the cutout part, and the one transferred in the donor's, the recipient's body will also regenerate into a complete liver, right? So that's what.
Ma'am, but in transplant, the whole organ is transplanted, right?
Yes, so this is only for liver. Unique case, let me erase all of this. Yeah, so this happens only in case of liver, right? Because liver has regeneration ability. Other organs, the complete organ is transplanted. You can't have half. But in liver, you can do this. So you will, so they have live liver transplants, which means the donor can also be
Donor can
alive.
also be alive.
Yes, recipient also can be alive. Similar case you can see in kidney also, right? But kidney is, you take out one entire kidney and give it, and you are left with one, the other has one, things like that. But in liver, it's just one organ which is cut. So they just take out a small part, less than 50% of it, and they transfer it into the recipient. Okay. This is a very interesting
Ma'am,
area of research. Yes. Tell me.
can growing of nails and hair be regeneration?
Uh, at some level, yeah, you could say that. But they don't really form an organ or any important part as such, right? Regeneration, when you look at it, it's usually something to do with an organ, or in case of starfish or lizard, it's an important movement part, right? Tail or an arm or locomotion part. So when it is those kind of things, we would call it regeneration. For hair and nails, I think you could term it more like just growing. Okay. Okay, yeah, so this is all about regeneration. Yeah, here they've given you a small note on how regeneration takes place. Right, so regeneration first is carried out by specialized cells. Okay, you have specialized cells to do that, right, which proliferate and make a large number of cells. From this mass of cells, different cells undergo changes to become various cell types and tissues. These changes take place in an organized sequence referred to as development, right? Okay. So there are specialized cells which carry out the process of regeneration, right? Proliferate basically means they divide and grow. Okay, another confusing term in this chapter and in heredity, or when you do when you talk about reproduction, is these two words. Okay, when do you say multiply and when do you say divide? It's very confusing, isn't it? Because some places they'll say the cells have multiplied, some places they say the cells divide. What is it? So basically you say divide when you get two daughter cells. That's what you call divide. When this process happens multiple times, you will call it multiply. Okay, when you have so many copies. Okay, so you'll have multiply. So basically proliferate means they grow or they divide and they grow is called proliferate. And make a large number of cells. Okay, and from there you will get different types of cells. This is the example of regeneration in planaria. Okay, now we have budding. Budding is, I think, the easiest one, right, among all of them. A bud develops as an outgrowth due to repeated cell division at one specific site, right? Because buds develop into tiny individuals, and when fully mature, they detach from the parent body and become new independent individuals. That's it you need to know about budding, right? This is the diagram of budding in Hydra. Again, for this also there is no specific location. Okay, for example, if this time a bud has formed here, next time the bud could form here, it could form here. There is no specific as a fixed location that the bud will be formed here only, right? What triggers it is the repeated cell division. So where the repeated cell division occurs is governed by enzymes and a lot of other factors, right? Okay. Yeah, so the bud will grow, and when fully mature, it will detach, and it is capable of continuing as a independent organism. Okay, and now we have vegetative propagation, right? It is a form of asexual plant reproduction. So in plants, you have both types of reproduction, right? You have sexual and asexual. Sexual would be observed in flowers. When flowers pollinate, that would be sexual, right? And asexual would be vegetative. Oh, let me not do this. And this is vegetative, right? Okay. Two types you have: natural vegetative propagation and artificial vegetative propagation. The difference between them is basically some of the plants, they have this inbuilt capability of propagating by themselves, like the figure shown here itself, bryophyllum, right? I don't know if you've seen the bryophyllum leaves. Has anybody seen them? They are actually quite widespread. I mean, you might see them growing
Yes.
in your housing area, etc. Might be a lot of them. Okay, if you haven't, I think you can just Google bryophyllum leaf. You might get a nice video where they show you how this actually happens. Okay, I didn't really check it. If you want, I'll find a video and I'll show it to you tomorrow. I'll do that. Okay. Yeah, so that is natural, where some of them already have this ability. Bryophyllum is an example. There are a lot of other plants which already do that, right? You could even Take strawberry as an example. Strawberry also has the ability of naturally propagating via leaves and adventitious roots, etc. Right? And second type would be artificial vegetative propagation. That would include all things like layering, grafting, tissue culture, etc. That's all artificial. That's when man intervenes and we try to mix and match and try to grow them by, or take out cuttings and try to grow them. Those would all be artificial, right? Yeah, they've given you some examples here. And these properties like layering, grafting are used to grow many plants like sugarcane, roses, grapes, etc. Again, vegetative propagation, like I said, it's a huge area where you have a lot of other methods also, but it's not required for you to know what are the methods. The only words which are seen in questions, or when you read this chapter you will come across, would be cutting, layering, grafting, and tissue culture. Only these 4 words you will come across when you read vegetative propagation. So all you need to know is they're all a part of vegetative propagation itself. Okay. Grafting is basically when you take, like I gave you the example of tomato, right? When you take a stalk or a stem of one plant, okay, let's call it plant 1, and you take, let's say, the shoot part or the tip part of plant 2, and you make a slit. Okay, you make a slit here and you push this in here. So this is called grafting. It's the simplest way I
It is
can.
common in mangoes also, right?
Is it? I'm not really sure. I'm not sure. I think it's very common in roses. That's how they got all those colorful roses also.
White flower, white rose, sorry.
All different. There's so many roses. That's how they got all of them, by doing grafting. Right? The blue, yellow, and white, and I don't know what other colors are there. There's some pink at the tips, but white inside, something like that also. Right? So this is basically grafting in short, in very simple terms. Right? Okay. Layering would be layering is another complex method wherein they layer one part over the other and they try to grow. Cutting is simple. Cutting is basically you cut the plant at a particular angle. Let me just draw it here. Right? Suppose this is your stem. Okay. So you will make a cut at a 45-degree angle, right? And then you just take out the tip, this top portion, and you plant it in soil or in water. Okay, and it will just continue to grow. That's called cutting. Yeah, this is mostly seen in sugarcane. Sugarcane grows from cuttings only. It doesn't really, there's no seed or any method of propagating it. You can use this for roses also. Roses work this way as well, right? And lot of other plants. You can take out cuttings of this commonly seen plants in the house. For example, mint, pudina, right? You might have seen. If you take those cuttings and grow, they'll also grow. You just need to make sure you have the nodes in your cutting and they'll grow, right? Lot of plants like that. Okay. So this becomes all part of vegetative propagation, and we have advantages of vegetative propagation. This used to be a very important question when they've asked subjective questions. I don't know how much weightage this would carry if you talk about just MCQs, right? But yeah, this used to be one question definite, used to just keep showing up in school exams, in all kinds of exams, would be what are the advantages of vegetative propagation, right? So I've just listed them down. Whatever is in bold is the keywords, right? So you just make sure you have those included, right? The 3 of them are: plants raised by vegetative propagation can bear flowers and fruits earlier than those produced from seeds, right? So you can call this a rapid method. Okay, we can propagate plants like banana, orange, rose, and jasmine that have lost the capacity to produce seeds, right? These are just some examples. Not all of them have lost, but banana in general, it's easier to grow them via vegetative propagation than remove those seeds and do it. Orange definitely has seeds. You can use seeds or you can use cuttings, etc. Right? Rose and jasmine, again, instead of waiting for the seeds, you can use cutting, layering, grafting. Okay. So it is a method to propagate plants that have lost the capacity to produce seeds. Another important one. And the third one is the plants. All plants produce are genetically similar enough to the parent plant to have all its characteristics. This is important when you are looking to preserve the characteristic of a plant. You don't want it to lose the important things. Like for example, you could take the example of plants which have certain vitamins or minerals already present in them, right? So when you don't want it to lose that, you can use something like this method. I can't really think of an example. Yeah, you could take maybe example of a spinach or any other leafy vegetable, right? So they are rich in magnesium and some other minerals as well, right? So when you want it to have that characteristic itself in a plant, when you want that magnesium richness in it, you can use to propagate plants like that. Or you can take the example of plants like basil. Okay, basil also is a good example. If you want that sweet taste of basil, you can use cuttings and continue to propagate basil so that you don't lose that flavor or you don't lose that smell. Okay. And these are some of the examples, right? Some of the examples of what parts of the plants are used for vegetative propagation. For example, you can use bud of potato, right? They are also called eyes, right? If you see a potato, they have these green spots, which, some green thing would show up at some spots. Those are called buds or they are called eyes also. Okay. So if you cut the potato, any part, including that eye, it will grow into a potato. You just cut it like this and plant this in the soil, it will grow into a potato. Right? You can do all these experiments, like all these plants would be there in the house, right? You can, and there would definitely be some soil around. So when you have time, just take a clipping and just grow it and see. You can use potato, you can use onion. These are maybe commonly found in your house, right? If you eat them. If you don't, you might be eating ginger, or you can take example of I gave you the example of mint, right? Pudina, that is commonly available. You can try all this. Even garlic also, you can use the bulb of the garlic to grow garlic if you're interested. You can do these small experiments in the lockdown, so you can, you have something to look forward to and you can keep taking care of it also, right? And then when it's ready, you can make some delicious dish out of it. Okay, this is just some example. Of course, there are a lot of other plants and lot of other ways to propagate them. So if you're interested, you can always pick up a plant of your choice and yeah, do it. Some of them can even be grown only in water also, right? So you can check out all that. Some fun activities you can take up. Okay, yeah. So bud of potato, rhizome of ginger, bulb of onion, right? Bulb would definitely be in this part. Oh God, what did I draw? Right. Okay. This is how the onion looks, right? So this is the bulb of the onion. Similarly, bulb of garlic also. You can take out one garlic clove and open the skin, remove the skin and just place it in water to grow. Okay. Leaf buds of bryophyllum we saw, adventitious roots of dahlia. So roots, whenever you see roots other than the place where it's supposed to form, which is at the bottom of the plant, they are called as adventitious roots. Okay. Rhizome of ginger looks something like this, right? It doesn't really have a shape. Just, yeah. So this part is called a rhizome of a ginger. So even this, if you cut and you grow, you can propagate ginger like that. Okay. So these are some examples. Of course, there are a lot many more. You can, you can search some more if you are interested, or you can, like I said, you can take up this as an experiment and just see how well it grows or what all you can do. Mix and match. You can also make your own lab inside the house, right? It doesn't take much space and time or energy, any of it. Okay. Okay, yeah. And now you have tissue culture. Tissue culture also, if you know this much is sufficient. This slide is sufficient for information on tissue culture. Okay, we've covered everything. We've covered the advantages and what is it and how do you do it also. Okay. So new plants are grown by removing tissue or separating cells from the tip of a plant, growing tip of a plant. Okay. These are then transferred into artificial nutrient medium where they divide rapidly to form a callus. So this undifferentiated mass of cells which forms, that is called a callus. Okay. That callus is then transferred to another medium containing hormones for growth and differentiation. And then they are placed into the soil so that they can grow into mature plants. Okay. So basically, for example, this is your plant, right? There is some plant, let's say mint leaves, mint plant. Okay. So what you do is you will take a clipping or you will take this much part of it, and usually this is done in test tubes. If it is something bigger, you could take a bigger flask or something like that. But usually it is done in test tubes because it's easier to observe the callus growth. Okay, and you place the medium in it here, and then you place this plant like this. You stick it inside that medium. Okay, medium is basically a solution, so it's like you could think of it as liquid food for the plant. But we add in solidifying agent. Usually we add in agar so that it solidifies, and then it becomes like a jelly, and you can just place this plant inside. Okay, and then you wait till this grows. Okay, and once it grows a little bit, you transfer it into the soil. Okay, that's how it works in short. Okay, I think I might have one picture of tissue culture. Let me check. If I have it, I'll show it to you tomorrow. When we had done tissue culture, I might have it. Okay, right. Okay, yeah, so this is how it works. And basically nutrient medium is, you can consider it as food, right? And you give it hormones and for growth and differentiation, right? Because you're controlling the environment, you provide whatever the plant needs for proper growth. Okay, and what are the advantages? This is important, right? What are the advantages is important. We can produce a large number of plants from a single plant in a disease-free environment, right? A majority of issue which farmers face currently, or which anybody who's growing plants commercially would face, would be diseases, right? There are a lot of pests, lot of microbes which harm plants. Okay, and which cause their yield to go down, and because of that your profit will suffer, right? You not get sufficient profit. So tissue culture is an expensive method. It's not like a free and easy and amazing method. It's expensive, but it will ensure that you have a lot of plants produced from a single plant in a disease free environment. This is the important point, right? And it's also used for the rapid production of ornamental plants like carnations, orchids, dahlia. Ornamental plants are plants basically which don't really have any value, but they're just used for show. Okay, show. So some examples of them are carnations, orchids, and dahlia. Right? To uh, you could use tissue culture to rapidly produce them. Right? Okay. Okay. Like I said, again, this is a vast field, and this is a really interesting field if you're, a person who likes tending to plants, or you're interested in, coming up with ways so that the plants don't really have diseases and things like that. It's a very interesting field of study. Okay. Okay, yeah. This slide, this I got it from, I think, one CBSE champion or some book. I'm not sure. I think CBSE champion only. So this is the uh, overall collection of all the points which are there for asexual reproduction, right? You can go through this later. We've covered all of it. But in case you know you want to do last minute studying and you want something, you can just open this and see. Okay. Okay. And we have, oh, I thought I had a video. Wait, let me see. Where did I put that video? I had a video for asexual reproduction. Let me just check. Ah, here it is. Okay.
As topic, you will be able to describe.
Is it audible? Is it fine? Can you see? Is it audible? I could.
Yes, ma'am.
Reproduction. Describe vegetative propagation and its types. Describe different methods of artificial propagation. Asexual reproduction is a form of reproduction that involves the production of a new organism by a single parent. Fission refers to the division of parent cells into two parts. This is the simplest type of reproduction and is seen in single-celled organisms such as amoeba and paramecium. In amoeba, the nucleus first divides into two equal parts. This is followed by the division of the cytoplasm. Thus, the two daughter amoeba formed grows and attain full size and then split again. In some organisms, such as Spirogyra and flatworm, the organism breaks up into two or more fragments after maturation. These fragments grow into new individuals. In budding, one or more new organisms grow in the form of a bulb-like projection called bud. It grows and then may eventually break away from the parent. If you view a solution of warm water, sugar, and yeast powder under the microscope, you will observe in yeast that a little cytoplasm accumulates at one end of the cell and the bud is formed. The nucleus divides into two and one of them enters the bud. In yeast, the buds form rapidly and create a chain of yeast cells if there is enough supply of nutrition. In hydra, bud appears on the body wall, which grows into a full hydra in a day or two and separates from the parent. A spore is a tiny spherical unicellular body covered with a thick wall. It grows into a new plant when conditions are favorable. Bread mold, that can often be seen growing on moist, stale bread, grows when spores settle on the bread. In the regeneration mode of reproduction, an entire new organism can grow from some pieces or cells of the parent organism. A flatworm can be cut into many pieces, and each piece will grow into a complete new flatworm. Vegetative reproduction refers to the production of new plants from parts of the mother plant, such as roots, stems, or leaves, without help of any reproductive organ. Potato. There are several buds present on the surface of a potato called eye. A piece of potato with at least one eye on its surface is planted in the soil to develop a new plant. Ginger. In ginger, new plants are produced by some parts of underground stems. Onion. In onion, new plants are produced from the bulb, which is an underground stem with thick leaves. Strawberry. In plants like strawberry, the shoots with the buds bend towards moist land and grows into a new plant. Bryophyllum. The plant of bryophyllum reproduces with the help of its leaves. A bryophyllum leaf contains various buds on its margin. When these buds fall on moist soil, they give rise to new plants. Several artificial methods are developed for vegetative reproduction, which are commonly used to grow many plants from one plant. This is referred to as artificial propagation. Some of these methods include grafting, cutting, layering, and tissue culture. In grafting, a twig of one plant called scion is kept over the cut stem of another plant called the stalk. Then they are tied up together. The tissue of the scion and stalk combine to form a new plant. This method helps in developing a high-yielding and disease-resistant species of plants. In cutting, a healthy young branch or stem of the plant having buds is cut off and planted in moist soil. The cutting develops roots and grows into a new plant. In layering, a young healthy branch is bent down towards the ground and covered with moist soil. After some time, roots develop from the covered part. This is called a layer. This branch can now be cut to form a new plant. In tissue culture, a piece of tissue is cut off from the growing tip of plants and kept in the nutrient medium under controlled conditions. The hormones present in the nutrient medium helps in the cell division. It leads to development of roots and shoots. These plantlets are then grown in the moist soil. Vegetative reproduction is very useful as it allows developing new plants very quickly, exact copies of the parent plant are produced, plants propagated with this method need less attention in their early stages of growth, and new variety of plants can be developed.
Right, this is all we discussed till now. It was like a recap, isn't it? Okay, yeah. There is one more video of fission also. Okay, what I'll do is I'll share this later, right? You can watch. Let me finish plant today, otherwise we might have. Okay. Oh no, actually we can do plant tomorrow also. What do you want to do? Do you want to see another video or should we do plant?
Miss, tomorrow is there a class on Thursday?
Oh yeah, Thursday. Sorry, Thursday. Should I finish plant reproduction as well, or should we watch videos? What would you like?
Watch video.
Watch videos. What about the others? You can say it's okay.
Yes, the human one is a big one, right?
Human one is a little bit big chunk.
If we could do the plant food, then fine.
Okay. What about the others? No response from you.
We could finish this part today itself.
Today, okay. Okay, let's finish it then. Chalo. Okay. Fine. We have something before this. Yes. Okay, so we'll move on to sexual reproduction, right? The production of offspring by the fusion of gametes. So here there are gametes involved, okay, usually from one male parent and another female parent to form a diploid zygote. Okay, another a few more new terms which you might come across. Zygote, though I think you might know, or if you don't, it's just a product, or what do you say? The end result of fertilization is a zygote. Okay. Haploid and diploid are two important terms which you may come across, or maybe you might come across next year if you take a bio game, right? Haploid basically means half, and diploid means double. Okay, if you want to remember the words in simple languages. Okay, so this refers to, it's usually terms referred or denote number of chromosomes. Okay, they denote number of chromosomes. Let me write it properly. Okay, when you say haploid gametes, okay, so gametes, when you talk about gametes, you talk about a single cell. Okay, single cell. So usually these are referred, or these are mentioned with reference to a single cell, right? You can't take a collection and then say this is haploid or diploid. Usually it would be for a single cell. Okay, and one more thing you need to know is, yeah, what is diploid and haploid and where they are formed, right? So this is diploid, let's say, and this is haploid. Okay, haploid, all gametes are haploids. All gamete cells, okay, whereas diploid is usually all other cells are diploid, all others, body cells, zygote, everything else is diploid. Haploid refers to 32 chromosomes, right? Okay, one second, I need to, yeah, the 32 and 46, right? Oh, I made a mistake. Yeah, that's what I thought. Sorry. It's not, this is reverse. Okay. Haploid refers to 23 chromosomes and diploid refers to 46. Okay. So each cell, each body cell, or each cell in our human body has 46 chromosomes. Okay. Total of 46 chromosomes, 46 chromosomes. What happens is, when the gametes form, they are formed via this process called meiosis. Okay. And meiosis, if you know, is called a reductional division. So the number of chromosomes gets halved. Okay, and so now you have something like this. Okay, so and then when fertilization occurs, one of this and one of this come together, right? Suppose this is male and this is female. Okay, I'm not writing it properly. I'll show you in the next slide. It's the animation and everything is there in that. Okay, suppose this is a male and this is a female. So 123M will fuse with 123F and will give rise to 46 celled zygote, right? Why is this done, if you would ask, is for this. If there were 46 and 46, when they fused together, they would have given rise to double, right? What is double of 46? 90 something, 92, right? They would give rise to 92 chromosomes. That would become error. That would lead to an error, right? You can't have that many chromosomes. You usually have 46 chromosomes. So what the body mechanism evolved to, or what it did is, only the cells which are involved in production of gametes, they undergo meiosis. All the other cells undergo mitosis. Okay, so that's why they are called, the cells are termed different, right? So all others here, let me use some better color. Yeah, they are what are called as somatic cells or body cells. Okay, whereas these are called gamete cells. That's how they are named and differentiate. Okay. So diploid zygote basically means diploid would refer to 46, right? So the haploid gametes come together to form a 46 numbered zygote. Again, this might not be asked that much right now. Maybe if you take up bio next year, this entire chapter, okay, I think in grade 12, yeah, grade 12, this entire chapter is broken down into, let me see, how many chapters? You have one chapter on reproductive health, you have one chapter on human reproduction, you have one specifically for flowering plants, whatever we are going to cover in 2 slides. There is one whole chapter on that, and you have a separate chapter for reproduction in other organisms. So that's like close to 4 to 5 chapters. This is broken down into. So you're getting the idea, right? So how much it is condensed and given to you right now. So there is just a brief gist of all the processes which is happening. You're not given in detail, so it's not limited to here. It's a huge process, right? So even when you do take this up further, or when you come across this chapter again in 12, they'll give you a little more information about the process, right? They won't dump everything on you. Okay, so slowly you build up information. As with every stage, you learn more about the same process. Okay, yeah. So that was about haploid and diploid words. Okay. So two gametes come together to form a zygote. Germ cells, which is small and motile, is usually called the male gamete. So basically, germ cells are the cells which produce gametes. Okay. Like somatic cells are the body cells. Right. Soma means body basically. Somatic cells are what are called as body cells, right? Similarly, germ cells are the ones which give rise to gametes. So the smaller germ cell, which is motile as well, or which motile means it can move. Okay, move, movement. It's able to move. Okay. So the germ cell, which is small and motile, is usually the male gamete, and the large germ cell, which usually will be fixed in one place and will have food storage or food reserves. It will be the female gamete. Okay. And sexual reproduction involves two major processes. What are the processes? First one would be the formation of gametes, right? Without that, you can't have fertilization, right? This is called fertilization. Right? Fusion of gametes. Meiosis, like I just said, it is a process. It is a reductional division. Do you remember we had done meiosis last year in grade 9? Does anybody remember? No. Okay. Think we did it in cell chapter, if you remember. So anyway, I took it up from there only. This is the recap of the division. Okay, I'll show you the video. Yeah, okay. Show you the video, or I'll just explain it. Maybe we can watch the video next class. Mitosis is also called equational division. Why? Because you get equal number of chromosomes, right? If you see here, they both have 46, 46. Whereas meiosis is called a reductional division. Mitosis is one step. Okay, this is two step. So the first step of meiosis is same like mitosis. This is, I'm just giving you a brief recap or a brief idea about it. Don't worry, they don't ask you so much in detail. At max, they might ask you what haploid and diploid is. So if you know that, that much is sufficient. And they might ask you which of them undergo meiosis, right? So meiosis is always for gametes. Remember that. Remember 23, 46, that's all. Okay. So meiosis happens in 2 steps. Step 1 is same as mitosis. If you see here, step 1 is same as mitosis. Step 2 you will get it broken down into 23.
Means how many cells are formed in meiosis?
Meiosis 4. Yeah, good you brought it to my notice. Correct. So here there are 2 daughter cells formed. That is also a difference. Thank you for pointing that out. And here 4 are formed. Okay. Okay. So what are the advantages of sexual reproduction, right? 2 main things. One which we already discussed, variation. So much we did in the introduction part, right? So that is the main advantage, right? And they will exhibit diversity of characters. The same thing they have given in a different term. So variation at different levels, basically. That's all. Diversity of characters would be there would be maybe some of them have better. Okay, if you were to take example of humans, right? Humans, we had different characters, right? If you take for example height, I'm taking only physical characters because I can explain physical characters using genes and DNA, etc. Right? Behavioral characteristics, they don't really have a direct connection to heredity. Okay, that's why I'm picking physical characteristics like height. Okay, some of them are very tall, some of us are short, right? That would be diverse characteristics. Then you could take eye color. This is good example because you get a lot of colors here, right? You have brown, which is the classic. You have black, which is rare. Blue, again rare. You have green. I think only 1% of the population has green eyes, right? And so on. So
Okay.
gray. Okay, yeah, gray also. Yellow also, I think. Right? So these become different characters. So now you have so many people or so many individuals with different characters like that. So this is one example. Second example is variation. Okay. These are the advantages. Okay. Now we can go to flowering plants. Okay, before we go to flowering plant, these are the 3 terms again which you might come across repeatedly. So I just jotted them down here. First, what are angiosperms? Angiosperms are basically it's a scientific term for flowering plants. Okay, they are called angiosperms. So not all plants produce flowers. Okay, some of them uh, don't produce flowers, and they mainly resort to other methods to continue their life or continue their species or to reproduce, right? So, and there are some plants which do produce flowers, so they are called angiosperms. Okay, and they produce flowers, and definitely if they produce flowers, they would be bearing seeds and fruits. Okay, and yeah.
Do angiosperms include the plants which produce fruits but not flowers?
Which plant? I mean, I'm not getting. So, in order to form a fruit, you need to have a flower, right? That's how the process works. So, which plant? If you tell me the plant, I'll tell you.
Like, do we have a flower for mango?
Do we have a flower for mango? Good question. No, mangoes form into a separate category. They are gymnosperms. They are not angiosperms. Okay, I got your question. So yeah, there are plants which form, they would be called adaptations or they are exceptions. So mango, which you picked up, is a classic exception in terms of a lot of things, in terms of a seed, in terms of
So
the.
what about orange and all? They also don't have flowers, right?
No, they do, they do have flowers. You have lemon flowers, etc. We don't see them, but they do have flowers. Okay. Yeah, there is a separate category of plants. So they are divided into two, angiosperms and gymnosperms, right? Gymnosperms don't produce flowers, and they lead to something which are called as modifications. So the fruit basically is a modification there. It's not a real fruit. That's why they are called false fruits. Mango is a classic example of a false fruit. Okay. Again, all this you will be studying in detail in 12th if you take it up. Like I said, this is a entire chapter. Okay. Sexual reproduction in flowers. In that you'll study what are gymnosperms, what are angiosperms, what is bisexual flower, unisexual flower, what are the different agents of pollination, how do they actually help, all those things. Okay. And how does the fertilization take place inside the ovary of the flower, right? Everything in detail. False fruits, multiple nucleuses, etc. All those things. Okay. Yeah. So for now, you just need to know these three terms. Basic, if you know, is sufficient. Bisexual flowers will be flowers which have both the male and female reproductive organs in the same flower. So now in flowers also, not all of them have both, right? So ones which have both are called bisexual, right? Examples would be hibiscus and mustard. Hibiscus you all must have seen, right? The red flower with uh, anther like this, right? It has red petals and a yellow stalk like thing coming out like this. Have you all seen it? Not seen it.
Yes, ma'am. I've seen.
See? Okay, wait, I'll just show it to you. This flower. Now do you all remember? This is hibiscus. Again, this is a very kind of place, must be there all around you. Loading it. Upset. Okay. Right? And you have reproductive organs are in separate flowers. The classic example is papaya tree or papaya flower. Okay, for papaya and watermelon. Okay. Okay. What we'll do is we'll just do these two slides. We'll do parts of flower and we'll do pollination. Then we'll continue on Thursday.
Does watermelon have a flower?
Yeah, it does. It does. It's what I
But
said.
when
We
we
haven't.
see it grow, it's like just a small fruit turning into a big fruit. There's no flower seen, right?
Yeah, they don't. We usually have not come across because I guess that's more to do with the fact that we stay in a city, right? We are not used to seeing flowers in so many things. Okay, but it does. It does definitely does have. Okay, parts of a flower. Yeah. So these are the basic parts of the flower which you need to be aware of. Huh. Stamen is the male reproductive part. This is the stamen, right? This part. Wait, this part is the stamen. Sorry. Okay. Yeah. It is made up of, it has two sub parts, which is basically anther and the filament. So this brown bilobe structure like this, which is present on top, is the anther. Okay. Yeah, they've given the description here. It is a bilobe structure. It contains these two brown thingies are basically pollen sacs, and they produce yellowish pollen grains. Right? If you get a chance, if you can maybe see a flower like hibiscus around you, you can just go and touch the anther. You will notice that you get some very sticky and a weird deposit on your hand, yellow colored deposit. Okay, that's basically the pollen. Be careful though, because some people do have pollen allergy also. So you might just start sneezing if you go too close to pollen. Okay. Yeah. So this is the male part, or this is also called the androecium. and carpel or pistil is called a female. Give me 1 second. Why am I confused with this spelling? Okay. Sorry. Yeah. Gynoecium. Okay, this is the correct spelling. Gynoecium. Andro. Okay. Andro basically refers to male, gyno is female. Okay, if you remember those two words, anything to do with gyno will refer to the female parts, anything to do with andro would be referring to the male part. Okay. Okay. Yeah. This is the female reproductive part. It has three components, like the stamen has anther and filament. Filament is not mentioned here because it's just a stalk. It doesn't really have any other function. So even if you don't remember, it's okay. I mean, remember any function of it is okay. Gynoecium has three parts: stigma, style, and ovary. Okay, you can remember SSO or SOS, anything. Stigma is this top part. Use another color. Okay. Stigma is this top part, right? It is basically its function is to receive the pollen grains. Then you have the style, which is the long part so that it gets transported inside, right? And the ovary is the most important part. It has the ovules and the egg cells and other accessories required to complete fertilization and turn into a fruit, right? So these are the basic parts of the flower. The diagram is also here. You don't need to know what a peduncle is. That is not of use to you. Receptacle also, if you don't know, is okay. Petals, these are the petals, right? The bright colored structures. And the green leaves, the green tiny leaves below the petals are what are called as sepals. Okay, yeah, this is also not required. This much is enough. Okay, and what is pollination is basically, this is the definition, it is the transfer of pollen grains from anther of the stamen to the stigma of a flower. It's called as pollination. Now this can happen in two ways. Either it goes from the anther of the same flower to the stigma of the same flower, that's called self-pollination, right? Or it can go from the anther of this same anther of one flower to the stigma of another flower. Okay, that's called cross-pollination. Now, cross-pollination, there are different types. Like you might have thought about it already. Maybe might have you might get this question when you think about it. So, what would you say if it is if I say there is a plant like this, there is a flower here, and there is a flower here, and what would you say if this anther moves to this stigma? This is also a type of cross-pollination. There are specific terms for this, which you might learn if you do bio later. There are specific terms. It's called something. Okay, yeah. This is also cross pollination. Whereas suppose there is a flower here, okay, and there is another flower, another tree here, and the anther from here goes to the anther, goes to the stigma to this flower. Okay, this is also cross pollination. Okay, both are valid cases. Okay. One thing to note here is the species or the plants will be the same species. Okay. Yeah. In very rare cases, the mixture would happen that it would be a different species or something like that. But the plants have mechanisms to reject pollen from other species. Right. So all these are cross pollination. Right. And how does this transfer occur is by various pollinating agents. Right. Pollinating agents are basically the helpers which help pollination, right? There could be a lot of agents. Common ones are wind, water, insects, animals. Humans could also be. When you accidentally touch one flower and then you touch another flower, you could also be helping them pollinate, right? Most important ones are bees, right? That's why they say we should save bees, because we would be left with no pollinating agents if you harm bees. so much accessible, right? Bees, wind, water, right? Some animals also, when they are searching for food, they accidentally touch their nose on the flower and then they touch another flower. So they are also transferring pollen grain, right? It's a very amazing thing in nature. You know, if you observe pollination, the most amazing thing. Yeah, quite fascinating as to how it works. And when you when you learn this further in 12th, they'll tell you how it rejects it also, like what all are the mechanisms or what all the plant has actually come up with so that it rejects pollen from different species, or if there is a mix match of pollen, how does it reject it, all that. Very nice. Okay, let us stop here, since yeah, let's do this tomorrow because I want to show you that video when we do it. Sorry, I mean on Thursday, whenever we have the next class, we'll do it. Okay. Yeah, I wanted to give you a couple of questions as homework. So when should I give it to you? Should I give you part by part, like give you homework for whatever we did till now today, and give you questions on Thursday for what we do, or should I give you together on Thursday? What do you
Part
prefer?
by part.
Part by part, okay. What about the others? I won't give you too many questions, don't worry. I'll just give you like maximum 10. Okay,
Part by part.
part by part.
But
Okay, everybody's cool with it. Okay, I'll give you it to you then. Okay. Okay, anybody has any questions or any doubts up to whatever we did today? Anything? Anything you didn't understand, or anything? Any questions? Okay. Okay. I hope, I mean, you all understood, right? Whatever we did till now. No issues in that. No. Okay. Okay, Alina said okay. 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 Anyway, the important part, this is all basics only. Go through it once more. Hopefully, read through your textbook or chapter notes once. And then let's see on Thursday's class. And of course, I'll give you questions also. No? Then on Thursday's class, if you still feel you've not understood anything, let me know. Okay. Okay. Okay, let's end the session then here. See you all on Thursday.
thank you, ma'am.
Bye.